Yo, what's up, baby birds? It's Tissue Tuesday, and today I thought we'd do something a little bit different and look at an individual cell through some transmission electron microscopy. And it's stuff like this that got me hooked on microscopy because if you look behind me, this is one cell. This structure right here, this big thing here, that's the nucleus of a single cell. This is actually a sperm cell from a lizard. And so when you get to see a nucleus this big, that's really exciting for people like me. So this is a nucleus. This up here, this is a structure known as the acrosome. And then this little black dot right here, this is known as the acrosomal granule. But when we look inside of the cell, what we can see is some of the organelles that are present, which allows us to make some hypotheses about what is happening inside of the cell. So up here, I can see the Golgi apparatus. This is the Golgi body. And I can see vesicles budding off of the Golgi, and they're actually fusing with the acrosome. So the Golgi is packaging content, and then it's getting delivered over into this acrosome structure here. Right? If we look over there, there's going to be some rough endoplasmic reticulum. There's actually going to be some rough endoplasmic reticulum behind me right here, right? So we know that this cell is making some protein. I know that it's metabolically active because right here, this is a mitochondria. The mitochondria are responsible for making ATP, right? I know that this cell is doing some stuff and getting rid of some trash or debris. This back here, if I lean back, this is going to be some cellular debris here. It's structures known as lamellar bodies, which are breakdowns of mitochondria, but then there's also some of all of this debris out here, and we've actually seen it happen where some of the membrane from the cell and from the acrosome during the condensate or during the process of germ cell development actually sheds some of that membrane, right, and packages everything down into a tighter compartment. And so this membrane gets shed as sort of extra debris. Right? And so by looking at the transmission electron microscope and looking at the individual cell, we can develop hypotheses about the physiology of the cell for some later tests. But if you want to look inside of a cell, you want to see a nucleus as big as your head, you got to do some transmission electron microscopy. Now you know. Later.